but it's really been a real honor, and I've really enjoyed this. And if I had they redo my slides after being at the conference, they would look different than they were. I had to do these in the week before, and my co-authors were trying to get me to get a paper done. And so it's a very quickly, and I probably have numerous misapprehensions, so point them out to me. Um, I would say though, years ago I was on the Health and Retirement Design Committee, and there's a guy named Richard Sussman, who was a major force in collecting the blood samples that were linked, and Kevin and Nick are using that now. He died in 2015, but he would be so pleased to see the work being done here. And I'd have to call this big science. I mean, this is really big questions with really state-of-the-art data, really large numbers of observations. So it's really, really impressive. Now, if I look at what I do, I would call it small science. I'm a little science guy right now. Part of it is just I have a little brain, and I can only look at I don't do macro even. But what I would say, we're talking about structural econometrics. I mean, maybe the closest I've had to a big science, I had a paper I published last year on the Danish automobile market. And where structural occurs for us is that we think of some things like preferences of consumers not being influenced by a policy like taxes. So what our paper shows is that Denmark has this incredible 180% tax on new cars. And it's what's called over the Laffer curve. It's so high that you could reduce the tax rate and increase tax revenue. And then you could rejigger the tax system and increase the fuel tax and make the new car tax zero, and it's a total win. There's less CO2, consumers are happier, and the government actually makes more. So that's a counterfactual. So why we do structural is partly to do counterfactual simulations, right? But the exercise I see here is more still, it's data mining, right? It's not guided very much by theory. It's much more of, there are some hypotheses out there, but you're looking for relationships. If I see the distinction of what I'm doing. So I'm going to here's an outline of, once you ask me to think big science, there's a danger because I quickly go off the reservation, but I probably won't get to these last 2 points. But I want to talk about the political risks that I see here. Because just Professor Liu's last comments where, the behavioral geneticists think everything's environmental. Whoa, that's really risky, right? And in this kind of politically charged environment. Now, on the other hand, I think that I'm totally against the kind of political correctness. Science should be allowed to be asking any questions, and if you don't like the answer, as long as you do it in a respectful way, you should be allowed to, ask the questions and have the discussion. Let me turn to that. I'm a former colleague of Arthur Goldberger, who Jim mentioned, and he was a reviewer of this book. How many people remember this book, The Bell Curve by Bernstein and Murray, right? And it was really controversial, I remember. And it's not a good example of science, I think, at least Art Goldberger, because they kind of it seemed like they had a preconceived message in advance. He said that their message was that intelligence is a highly heritable trait, plays a critical role in socioeconomic achievement and social pathology, and is becoming increasingly unequally distributed. This leads to an increasingly stratified society, a trend which compensatory interventions cannot halt, right? So it's got to say, throw up your hands, you just got to accept this. And I think Jim's work would be very diametrically opposite this kind of message. And Goldberger said that this really wasn't science. It was like a big publishing rollout where it hadn't been pre-reviewed. And, for Art, this is really cutting. He says, we know no more after studying the bell curve than we did before. So I don't think there's any danger of that being done here, but there's got to be sensitivity about, the role, because, there are really racist, there's congressmen who are, fairly racist. And if you say genetics is a big effect, they say, sure, that's exactly what I thought all along. There's no point. We can just drop food stamps and we can drop early education funding. And, that would be, I think that could be damaging. Now, that doesn't mean we shouldn't do the science. Now, personally, I think genetics plays a very big role. but it's most evident when you go back at evolutionary timescale. So I'm a kind of a, here again, I'm going off the reservation, amateur anthropologist, but I'll just cite some of the work that I've looked at. And, the basic message is that brain size has been increasing through history and the bigger brains are winning out. And Homo sapiens, why are we dominating Earth? It's because of a bunch of mutations that led us to have bigger brains. And that's just so clear that the genetics had a big role, a major role in why we are dominant, why we have the higher generalized ability. And so this is from the Smithsonian site. It says large complex brains can process a lot more information. That was a big advantage to early humans in their social interactions and encounters with unfamiliar habitats. So now here's a picture from that site. There's the Homo sapiens brain, and our immediate predecessors are called Homo erectus. It died out about 100,000 years ago. I think the last bones they found were in Java somewhere. But our brain is substantially bigger. And what are the defining features? That cortex, the outside part of the brain, which is more of the logic, and it's kind of mediating the inside, is more the primitive knee-jerk emotional brain. And that's been where most of the rapid evolution from my reading of this literature. Now, if you go back, according to I'm going to skip this slide, but this is from the Smithsonian slide that kind of argues that more environmental change quickened the path of the increase in brain size. But I think that correlation isn't as clear as I would like. But there's something called, what is it about that separated us from Homo erectus and the early people 70,000 years ago and spreading out all over the world? And so here I'm quoting from this work by Yuval Harari called Sapiens, which I think is a pretty cool book, although it's controversial. It's, they call it the cognitive revolution. So what caused this split that made us so superior? And, they define something called a tree of knowledge mutation. And the question is how many mutations are there that led to humans being Homo sapiens being so superior? And part of it is language is really clear, and from what I understand, language evolved around 150 spoken language 150,000 years ago. And it's that communication, the social environment that was really, really powerful that other animals don't have. But that's not the only one, but this is one that Harari emphasizes. But then there's been, I was looking at Nature in September 2022, new results, it's an article quoting another journal, Science, that 500,000 years ago, they found a pivotal genetic mutation which caused their brains to improve suddenly. And they said that the number of brain cells in the hominins that preceded modern humans gave them a cognitive advantage over their Neanderthal cousins. Then I saw another study by University of Barcelona, and it made an estimation of the time when some of the genetic variants that characterize our species emerged. And it did that, that's kind of amazing. You're looking at mutations that occurred 500,000 years in the past. We're struggling now looking at the present, but how do they do this, and is that credible? But it says that they're analyzing mutations that are very frequent in modern human populations, but not in these other species of archaic humans. So they have some old DNA, and they can compare the DNA apparently. But then they can actually date this, and they say that there's two key ones. One is about 40,000 years ago, associated with the growth of Homo sapiens in departure from Africa, and another one about 100,000 years ago, and was the time of the greatest diversity of types of Homo sapiens in Africa. So anyway, that and then there's another article this Nature article that actually identifies a gene called TKTL1. Does anyone know what that gene is? But it makes a protein that it encodes a protein that's made in the fetus's brain when it's first developing, and a mutation caused to change one amino acid resulting in a protein that's different from those found in hominid ancestors, Neanderthals, and non-human primates. So they said that this could have led to bigger brain size, and they actually did an experiment. So they injected this TKTL1 in the brains of mouse and ferret embryos, and found that it had significantly increased their neural progenitor cells. So that's really really cool, very powerful in some sense. So so what when I look at all that, I say undoubtedly genetics is really, really important. Now, the other thing though is that society, are we so smart because our brains have just gotten exponentially more smart than basically hunter gatherers? And I think there's a lot of evidence that we're smart because of mass cooperation. It's not that we're just that much more smart. And it's kind of like each neuron over time hasn't gotten much more functional. What's got is the more neurons in the brain, and they cooperate, your brain can have this epiphenomena of consciousness and rationality, right? So societies are the same way. It's like we're individual cells. We're not getting that much better, but we're interacting in societies which are sharing information and allowing specialization, division of labor, and where just our science, our collective culture is really performing. And so this is mass cooperation. I like this quote here from an anthropologist, Kim Hill. Humans are not special because of their big brains. That's not the reason we can build rocket ships. No individual can. We have rockets because 10,000 individuals can cooperate in producing information. So I think, and so then if you look at why did we cooperate? Like if you look at apparently chimpanzees, they're very tribal, they have very small groups, and they fight. You have two different tribes, they kill each other. Whereas the part of this tree of knowledge mutation might have been that humans at some point establishing pair bonds between male and female and offspring and next of kin. And the argument, there's a guy named Chappé, Bernard Chappé, human kinship, that this is what's unique. One of the factors that makes unique about humans is that we can have larger cooperation, and that led to villages and towns and societies where we're getting the real benefits of mass cooperation along with language. So anyway, I think you can say, wow, in some sense, you look at that, that's pretty impressive to me that genetics is really important. But now in honor of Jim Heckman, I want to come to the present now, and more topical is what I would call evolutionary selection bias. So, why is it, if it's so obvious and, when you look back in time, genetics is so important. In a present time, there's a lot of different numbers when you look at the literature, and they become much smaller, the role of genetics versus environment. And I might describe this, I don't know if there's a word that you guys have, but in honor of Jim, I call it evolutionary selection bias, which is that we're all kind of selected through, we're the result of millions of years of evolution. And, we have this common ancestor, Lucy, I guess, right? And we have 99.6% of our DNA in common. That means there's only 0.4% of our DNA that's different. So we're basically mass-produced copies of each other. So we shouldn't expect us, if I was comparing myself to a Homo erectus, there would be a huge difference in cognitive abilities. But I compare myself to, a lot of other people in this room. Maybe I'm a little bit lower, but we're not that different, right? So, the question is then is in this 4%. There's a lot of genes in this, a lot of SNPs in this 0.4%. But the question, and some of them is obvious. It affects hair color, sex, and various inherited diseases. I have one called von Willebrand's disease. It's very clearly identified in my genes, and it causes me problems, bleeding problems, and so there's definitely a role for genetics. But the problem is that when you look at ability, first of all, we're hampered because how do you measure ability? You're using something that's supposedly objective, like educational attainment. But, I don't want to insult cosmetologists, but if you got a PhD in cosmetology, is that equivalent to a PhD in mathematics? So, we're trying to use simple numbers, but there's all kinds of abilities. Jim's work and others that talk about, different cognitive ability, but there's social ability, there's artistic ability, creative ability, right? And we're very different. And we try to just line us up on just one dimensional metric. And maybe clearly some people will not stack up well on certain metrics of cognitive ability, but excel in other measures of ability. And so, in some sense, we're stacking things. Um, so, um, basically, I remember this. The line. Well, it's 50% success in life is 50% genetics and 50% environment, right? And that's the number that sticks in my head. But then I'm reading on these twin studies. That's not that far off from what they actually are getting, right? When I look at them. Um, uh, so let me show you this one, and maybe you've seen this one: genetic environmental variation in educational attainment, an individual-based analysis of 28 twin cohorts. They're using 193,518 twins, and information on educational attainment 25 years at 25 years of age and older. Now, what they use is what this thing called the ACE model, right? Where the variation in some quality like education is there's a genetic component which they call A, a common environmental called C, and then there's this residual idiosyncratic environment. And what they're coming up is A squared. I guess that's the fraction of variation due to the genetic, 43%. And then C, the common environment of the twins, that's 31%. So that only leaves 24% left for idiosyncratic environmental. That's not different, that much different from what I thought was kind of obviously BS, 50%, 50%. Now, the problem is that how do you separate? I don't know enough about these. You got common genes in a common environment. They seem very serially correlated, like collinear in a regression. How do you separate the part that's, like if you have really rich parents, they have twins, maybe I got good genes from my parents, right? But I also got a really great household and they were highly educated. They helped me get into the world. How do I separate what part is genetics and what part is environmental? Now, the ideal experiment would take twins and at birth, and then randomly assign one of the twins, like send your twin daughters, send one of the daughters to grow up in Afghanistan, right? And one gets to grow up in Beverly Hills and in Afghanistan. Well, that would be a really cool experiment. I don't think it's going to pass human subjects review, but we have something close, right? I think there was a 1990 Minnesota Twins experiment, right? Does people know about that? And they looked at twins separated at birth. It's a guy, forgot the name, but it was published in 1990, and they looked at IQ, and among the twins separated at birth, they ascribed 70% of the variation in IQ to be genetic, because they had different, very different environments. They were raised separately, but they could account for 70%. So that's even bigger than 50% from that study. Which now I don't know how much it's been correlated with other studies since then, but these are big numbers. Now let's turn to GWAS. Now there you get lower numbers, and the question is why. The numbers seem to be lower to me. So yeah, this is really impressive study. Some of you right here are part of the, Ben, you're part of the authors. So several people are authors of this, right? It's Akbai is the lead author. And first of all, it amazes me how many co-authors you have. We have trouble as economists policing 3 or 4 co-authors and getting them to do their, there's like massive shirking. Like when you have 50 co-authors, how do you get to make sure everyone, who's the lead author who has to make sure they're doing their work? I mean, that just amazes me right there. But, that's a side comment. But this paper, it's got 3 million individuals. But now if I read it right, the PGI index that they created explains 12 to 16% of EA variance. and contributes to risk prediction for 10 diseases. So that's considered a success. But why only 12 to 16% when the twin studies were so much higher? Now, is there something that that we have probably inferential problems in constructing these PGI index, and they're actually higher, but this is measurement error or something, attenuation bias? But and you would expect they should be higher because in the twin studies, you got this collinearity between environment, but in these 3 million, you got very different environments. So maybe that's what's going on, is the environmental variation is much bigger than the twin studies, and it leaves less for genetics. But I don't know if people have kind of tried to relate those two and say if there's anything inconsistent. Now, one of the things that it was Professor Liu and I think and Professor Lee were talking about is the direct effect of genetics versus through your parents, right? That so I think they're controlling for parental PGI as well as the individual DDPGI, and so they find that when for educational attainment, once you control for the parent, then it even falls less. So the direct effect of it. But then you're coming down with issues of causality. You might say, I'm giving some genes to my daughter, but she's getting a little bit different. How much is her success or lack of success due to her variation from the parental DNA? And so Jim raised that question, and well, I think we've raised some different issues, and our direct effect, indirect effects of genetics, the effect of the environment. This is this language is a little bit confusing to me. So now I'm also find it this linkage disequilibrium hard to understand. It's something about correlation. Yeah, I get it's first of all language linkage disequilibrium. I don't know what's disequilibrium, but I guess it's if you're nearby SNPs are generally more correlated, but not always, right? So when I understand that that's not happening, and then there's something about you're adjusting the intercept because of pretest bias. So this is from that paper I just quoted, and I guess using the intercept and how much bigger than it is one, but it's a little bit murky. Like in econometrics, we are like highly theoretical. I'm not saying it's good necessarily, but we have we talk about consistent estimators and count instrumental variables and really rigorous theoretical study as well as empirical work. A lot of this seems to be a little bit ad hoc to me. I see simulation studies, not maybe I'm missing it, but I went back to this and further, when I look at the studies, there's a scatter plot of here's the educational attainment from Ad Health and HRS, and and apparently this this doesn't even look like a 16% R squared. When you look at that level of variation, it looks like the PGI is explaining very little bit. And there's some other structure. I mean, you have PGIs around here, and then you have highly educated having about the same index. And I'm thinking, is this really a good prediction or not? But then at the same, just after that, there's this graph. And I look at that, it looks to me, if you take PGI decile, it looks like it's a completely strong prediction. So I'm saying, are these both from the same data set? They don't compute to me. This figure here looks like a really weak, association. This makes it look like it's a very strong. Maybe you have an explanation, but when I read it, I have all these questions. There's an article by Ware in BioRex IV or something. What the name of that journal is. But they basically talk about that. So you're running these millions of univariate regressions, and there's we wouldn't even call that stepwise regression in econometrics. It's like running a bunch of them, seeing which ones are significant and which ones have a p-value bigger than 1 to select a small subset. And I think it raises a lot of issues about pretest bias and so on that I think econometricians would be concerned about. And I guess that's the linkage disequilibrium is designed to address those issues. But to skip, they've been talking about, so there's been, I found many papers that are, critiquing this whole methodology, and I don't know how controversial it is in this literature. And another paper in Annals of Applied Statistics that they say further SNPs included in PGSs are often chosen using discovery thresholds based on p-values, which are known for their far-reaching limitations. And final PGSs are obtained using somewhat arbitrary weighting of the SNPs. So I don't know if those are fighting words. I'm just blundering into this. But when I look at machine learning, it seems to be very, bypass many of these problems. So there's we talk about epistasis, which is potential nonlinear interactions between that Jean-Pierre raised, right? Why is it just this linear index? What couldn't there be these really complicated interactions that could be discovered? And then there's also many right-hand side variables. Well, the machine learning methods can handle a lot of that can go. Now, maybe it's just, so, that's what lasso is. And yeah, here's the lasso, and then elastic nets is a version of lasso where, you're changing the penalty, whether it's another parameter, alpha, either a squared penalty or an absolute value penalty. And what these things do is you just throw everything in the regression and it just selects a subset that's going to be significant. So you don't need to do this first stage. It just will identify the subset. And there's some very favorable, Viktor Chernozhukov and others have talked about the post lasso. You do a first stage lasso to kind of identify the model, and then you can do a second stage regression with the selected model and trust the standard errors and the statistical properties of that. So, and so some of these studies are finding very big increases in prediction in prediction accuracy from using these lasso approaches. So I guess the question is what what do you think about that? Why aren't you using that approach? Now, then finally, there's deep neural nets. Everyone's talking about that because I saw an article in the New York Times, deep neural nets for mammograms, right? It does better than humans. And the protein folding, this amazing predictor of, it's called AlphaFold, that was developed by DeepMind at Google to predict protein folding. It's just like a complete breakthrough in science. So deep neural nets in chess, we know, and so many other things, it can handle many interactions and discover deeper structure. So the question is, and so I found an article on this by some people in China, a deep learning genome polygenic risk score for common diseases identifies individuals with risk. And they're claiming this is pretty capable of discovering and producing better predictions. So they're using more traditional SNP selection, as I can see, to create the input SNPs, but then using these deeper layers to actually get the better predictions. And they call it the deep polygenic risk score. So this allows all sorts of interactions. That's what deep neural nets are good at. So that, and in principle, you wouldn't even have to have this one. If you have more layers, you just throw everything in there. And deep neural nets, you get a huge number of parameters. So I guess it's the total number of parameters that you can really estimate with a data set. But in principle, if you have enough observations, the deep neural net just totally discovers the structure. Or at least that's what's alleged. So, okay, so the last point I'd like to conclude on, though, is, all right, there's methodological things, and we're discovering some very cool things, and there'll be battles about methodology for a long time. But also the goal, when we think about the big science, there's a criticism. You could say, well, it's heroic, and that's not always a good thing. A heroic science means you are venturing into territory where there's so many unknowns and you're just finding crude correlation, but you don't really understand it. But when you look at other areas like health, where it shows tremendous understanding, these are the genetic cures, right? I just saw on the NIH website, I have von Willebrand's disease, but there's hemophilia type B. They just, have a genetic treatment for that, where they inject messenger RNA, goes into the epithelial cells and edits. And so this thing called CRISPR, this amazing thing, they can just basically go in and genetic edit in vivo in people and cure a number of diseases. So that, not only you're identifying the specific genes, but you're figuring out how to go in and fix them. That's totally amazing to me. So, and in a general, you might say there's ethical issues, but when you're fixing diseases like some of these sickle cell anemia or hemophilia, you'd say, who can object to that? So there's amazing advances, but in things about inequality, I don't know how the prospect is for policy to change when these are so politically divided issues. So I'll just stop there with that point.
Points and questions that John raised that I think are useful. I mean, I will totally take questions, but I wrote down a few things where I think there are experts in the room that can provide some clarity and just show us where the frontier is. So one thing that John highlighted or mentioned was this missing heritability problem, this kind of gap between the estimates of heritability that we get from twin studies and other designs, and then these sort of SNP-based approaches. And so I think this would be a very good place where the people in the room who are experts can talk a bit more about that. I know James, you mentioned some of the reasons. James, you maybe want to respond to that.
Sure. So at a first cut, we can divide methods to estimate the heritability, which is fraction of the trait variance due to genetic differences, with either old-fashioned or classical methods based on, well, the correlations between relatives, twins, and so on. But then now we can do it with DNA-based methods. One important thing to note is that even with the family-based methods, different designs actually estimate different heritabilities. So if you're looking at what my predecessor in Minnesota did, Tom Bouchard, with the Minnesota study of twins reared apart, that is estimating something we call the broad sense heritability, which includes variance due to interactions between genes at the same SNP, which we call dominance, between genes at different SNPs, which we call epistasis. Also, what they call the classical twin method, which is basically dividing the dizygotic, subtracting the dizygotic correlation for the monozygotic and multiplying by 2. You can do that with maximum likelihood, but that's basically what that is. This also contains a component due to this non-additive. In fact, that component can often be inflated. So when you hear a number like, well, the correlation between twins reared apart in IQ or BMI is like 0.7 or 8, that's an estimate of the so-called broad sense heritability. Certain family designs, however, do not include that non-additive component in its estimate, or less of it, such as, for example, the correlation between parents and their own. As I was saying, basically any kinship that is not monozygotic twins will contain a severe attenuation of the contribution to their correlation from non-additive components of the genetic architecture because of the breakup of these multi-step configurations due to recombination and independent segregation. And it is true that when we look at estimates of heritability from, say, parent-offspring regression as opposed to twins, whether it's twins reared apart or the classical twin method, they do tend to be smaller. For example, the heritability of height estimated with twin methods, often you can get numbers like 0.9. But when you do parent-offspring regression, it's more like 2/3 or 0.7. And you see similar attenuations with IQ, BMI, and so on. Then we move on to the DNA-based methods, which for the most part are only estimating the narrow sense heritability. That is the heritability due to only the additive component. It turns out it is possible to effect an orthogonal decomposition between the additive component and the non-additive. So it is possible to extend these DNA-based methods, we call them GREML and so on, as Dan was saying, to estimate the non-additive components. And as he said, so far, the numbers we get back are all negligible. But just concentrating on the additive components for a moment, there's this question of why do these not approach numbers that we might have heard of from twin studies? Well, part of the answer is that twin studies are estimating a slightly different quantity, the broad sense heritability. So there's no way GWAS polygenic scores or additive estimates from GREML can converge on that. Second of all, most of these estimates are derived from just common SNPs. That is SNPs where both alleles, or one of the two alleles, has a frequency above some cutoff, which is often set at 0.05, 0.01. There are many reasons for that, which include the low statistical power to detect associations at rare SNPs that fall below that threshold. And also the fact that often we're using genotyping chips, and genotyping chips cannot be used to impute rare SNPs very well. So basically, we're estimating heritability with DNA-based methods and polygenic scores and so on from a subset of all SNPs. So from right there, we would expect the estimates to be smaller. Now, you might be thinking, well, I mean, common SNPs, if we think of allele frequencies as ranging from 0 to 0.5, and you define a rare SNP as anything below 0.01, surely you should get most of it with 98% of that interval, right? But it turns out that that is not the case because natural selection tends not to allow variants of large effect to become common. So basically, variants of larger effect tend to be concentrated at lower allele frequencies. So therefore, that 2% interval should actually punch above its weight in its contribution to the heritability. So we do certain kinds of studies, whole genome sequencing studies, for example, where we are no longer limited to just the common SNPs. We focus on the vast majority of the time in GWAS. And then we can apply these DNA-based methods like GREML to the data we get from those studies and see if now are we converging on the narrow sense heritability that we estimate in certain kinds of family designs, such as parent offspring regression. For certain traits, that seems to be true. For example, height. A whole genome sequencing study of height has come up with an estimate of the heritability of height that I think is 0.7, which is spot on close to that 2/3 0.7 number I mentioned earlier from parent offspring regression. One of my colleagues, Scott Vries, who specializes in substance use, he's just come out with a paper with doing basically the same thing, just rolling in the contribution from rare variants to common variants and comparing that to these pedigree estimates of heritability. His standard errors are big, but also he gets, at least within statistical resolution at present, consistent estimates for those two designs. The family-based heritabilities and the GREML herd. There's certain other traits, psychiatric disorders and so on. BMI is actually the best example, probably, where it looks like actually, I don't know what the narrow sense heritability estimates of BMI are, but certainly the twin-based estimates, for example, twins reared apart, both in the US and in China, show a correlation of 0.7, 0.8. Even with whole genome sequencing, we are not getting a heritability anything close to that. We're getting more like 0.3. So I would say that it depends on the trait. Some trait looks like, as we add in more variants, we will get close. I mean, if it's like 0.65 and 0.55, I'm like, whatever. But
So Jonathan, do you want to jump in here with something?
Oh, no, I agree with everything you said. I'll just give an example from the study of genetics of educational attainment. So in 2013 was the first US educational attainment with 100,000 people, 100, and the R-squared of the polygenic score there was 1%. Then 2014 was 300,000, 2016 was 100,000, 300,000, it was 3%. And then with the A4, with 1 million people, it went up to 10% or 12%, I think. And then the last one, the A4, is at what, 13% or somewhere around there? 14, okay. 15, okay. So the point is, there's two main reasons. The first is like we don't capture the whole genome variants, we just take the common steps, so that we don't capture the whole genome in the first place. You can expect to explain the whole heritability. And the other thing is that the beta is estimated with noise, but that's the reason why, as we get larger and larger GWAS, we get more and more precise betas, and that's why the scores has been improving. But
no, I have a third factor, which is that we're analyzing data from cohorts, but as we discussed yesterday, the twin betas are not exactly the same across cohorts. So if the heritability of education is 40% in Sweden and Norway, and you make analyze two twin studies, you end up with something with close to 40% expectation. It could be that the betas that generate that 40%, the variance are not exactly the same. That's something you can estimate and look at in the data, and that's source of attenuation here. And we have some reasonable estimates of what these cross-cohort betas are on average, right? And what do we think they are? 0.7, 0.75, something like that. So that's not a major factor. At least for expectation.
So there's one question I'd like to raise here, just thinking of why that, what impact that has for the kinds of analyses that applied social scientists are doing with these polygenic scores. It may not matter if, say, the common variants that are being picked up in polygenic scores that we currently have are behaving the same way, right? That a polygenic score that included the rare variants would, right? So if like your economic question is, I want to understand the mechanisms that would go from genes to informing education, for example, to affecting education, if the polygenic score based on common variants is going to tell me things like, oh, it loads on this much on cognition or this much on non-cognitive skills, etc., then I might not care, and I would if it didn't. So I guess a question for the statistical genetics experts in the room is, what do we know about, as we move to rare and rarer variants? For example, you could imagine in the enrichment studies, right? Are there systematic? Do there tend to be systematic differences in the gene sets that are enriched when you move to the rare variants that have large correlations opposed to others? I mean, is that fair?
I have a good comment. I don't think enough has been done on educational attainment, but I know a bit about autism, which is genetically very correlated with educational attainment. The answer is that's not true. So there can be very systematic differences between the rare variant signals and common GWAS signals for autism. If you look at the exome sequencing studies where focus on extremely rare variants for autism, it's very well understood biology. There are about 110 genes identified for autism. They're enriched in very specific pathways. For example, chromatin modifier. These are the switches that decide what part of the genome is open or closed. If you get hit by rare pathogenic variants in those important genes, you get comorbid neurodevelopmental problems, cardiac developmental problems, and intellectual disability, very important. But then if you look at the GWAS for autism, right? First of all, those genes don't get hit, don't show up as your GWAS thing, which is a mystery itself. But second, if you quantify the shared genetics between autism and educational attainment, you get very substantial but positive genetic correlation. So this is saying the genetic risk for autism is correlated with higher cognition. There, in rare variant, it's a negative correlation, intellectual disability comorbid with autism. So you really need to be careful with this. But again, whether you can port this type of conclusion to EA as a phenotype is a question mark. But you just can't assume it is always going to be the same.
So again, this might depend on the trait. So there was a paper that just came out in Nature where they examined exactly this question about which gene sets are enriched by common steps versus rare variants. And I don't think they looked at behavioral traits, things like height and so on, but the things they looked at and the abstract, they say that the enrichment patterns look similar.
I think that I'm digging myself in a hole because we're a little bit over time, but I also think there's interesting questions that are broad and kind of interest. I'll save it for later. Sorry. I think it's we've gone over our 10-minute discussion here, so I think next up we'll have Ben presenting.
Going after these great talks by John and James and Q is kind of a tough task. So this is the title of the session, not sort of a description of what I'm going to talk about. I'm going to talk about something a little bit more narrow, which is sort of how I think about measurement error in the PGS scores. I guess that's redundant. The PGSs. Just a little bit about my background. I'm an economist. I'm a professor of economics at George Washington University, and most of my research is in factor models and item response models for test scores and psychometric traits, and how sort of to think about incorporating that kind of data into economic models of skill formation and things like that. And I have other work on estimation and identification and panel data models. So so I don't have expertise in genetics. This is so everything I'm going to talk about referencing genetics is just what I've learned over the past month in preparing for this. So hopefully what I've learned is not already outdated, but I'm starting to get the sense that some of it is. But I think what I want to talk about is a few I think helpful ways of thinking about the measurement error, if we want to call it that, in the PGI or PGS. And so I'll talk about three aspects of this, right? So I think it's important. One thing I've learned from my from thinking about these models of test scores and measurement error and test scores is that the bias that this leads to matters. What bias this leads to depends on what we're using the score for, right? So if we're using it to estimate heritability, or if we're using it as a control variable in a regression or a structural model, or if we're trying to estimate G by E effects. So let's sort of talk about those three separate issues. Then I'll talk a little bit, one or two slides I think at the end of this, about sort of how these PGIs sort of compare to test scores, or how we aggregate across items on a psychrometric scale. So often we'll take a weighted average of items on a test or a Likert scale or something and throw that into a regression. And that's a problem I've thought a lot about. But so in some ways it looks like a PGI, but if we don't get there, the punchline is that I think it's a very different problem, and so maybe it's not useful to think about the methods that are used there in the context of PGIs. So I'll start just thinking about this issue of heritability and estimating heritability using genetic data. So this is sort of simple model for phenotype Y, which is G plus U. So G is the genetic component, which is this additive sum of the SNPs, X, Xij, and everything else U. Right. So I'm not sort of assuming necessarily that we don't have additional controls in the model. Just giving sort of, we can think about maybe we have additional controls in the model. So I'm going to assume, for instance, that this G is uncorrelated with U, right? Maybe that's after conditioning on a bunch of things like the principal components of the genetic data, or family fixed effects, or so it's the results I think can handle that. So I'm going to assume that, and then I'm going to assume that however we construct this, right, just to start, however we construct this PGS, this G tilde, right, so it doesn't even have to be a PGS. So I'm just going to start with saying we have some measurement of G, right, some estimate of G. So it's, and that's what I'm calling G tilde. And that's also uncorrelated with the error term, at least after we've sufficiently controlled for things. Then sort of the basic result is the heritability parameter we would estimate using G tilde instead of G is going to be the true heritability H squared times this correlation between G and G tilde, right, which deflates our estimated heritability. So no matter the nature of that G tilde, how we construct it, right, we're going to get this deflation, this bias downwards, and or attenuation bias in the estimate of the heritability. So we can think about some examples then. So if G tilde is estimated from a PGS, from a GWAS, estimated as a polygenic index from a GWAS, then I sort of, my understanding is that one case for the measurement error not being substantial is if N is large in the primary study with the GWAS study, and sufficient controls to control for things like what's it called? Geneticists have these names for things that we have different names for in econometrics. I keep forgetting the names, but population stratification, that's what I'm looking for. So control for things like population stratification and so on. And the correlation among the X's, so the linkage disequilibrium, if it's sort of limited to clusters, then we're able to sort of pick the right causal variance based on some criteria. So this is the one that I think is more problematic, and so I'll focus on it. But I think there's a sort of theoretical case for if we do this the right way, then we can pick out the right SNPs, and we don't get measurement error. if the linkage disequilibrium is not too, right? So that picture that James showed suggests that most of these correlations are zero. That sort of makes it seem like maybe the LD sort of issue is something that we could solve by doing that the right way if our sample size is large enough. But so what I'm going to do is I'm going to think about mostly that last problem. So what if that's not the case? And we construct this PGI score by doing these univariate regressions of y on each SNP, each xj. We get some estimate beta j hat, right, from the GWAS. And we're gonna so I'm assuming that the xj's are uncorrelated with u as well. This is in the secondary sample, not in the GWAS. Now, in the secondary sample, right? So the sample that we're going to use to estimate heritability. And the x's have been standardized, so they have variance 1, just to make the formula simpler. And I'm going to think about sort of the bias conditional on the GWAS sample. So sort of condition. So the beta hats are estimates from this initial sample. So you could either think about this as like n is large enough that we are ignoring the sampling error, or just think about sort of conditional on whatever those beta hats. Whatever the not conditional on beta hat, but conditional on the parameters that are conditional on the sample, conditional on the initial sample is what I mean to say. So in other words, the beta hats are going to have sampling error, but they're also going to have bias, and I'm focusing on the bias. Right? So they're going to have bias because the x's are correlated. If the xj is uncorrelated with all the other xk's, then we don't get any omitted variables bias. But generally there is going to be some due to linkage disequilibrium. So we want to think about that correlation. So this is just a simple sort of omitted variables bias formula, right? In the limit, as the sample size in the GWAS goes to infinity, the probability limit is just going to be this weighted sum of the correlations, right? So so it's going to be psi j plus an omitted variables bias, right? Psi j is in there multiplied by the correlation between j and j, which is 1. So this is just a condensed way of writing that we get thing we're trying to estimate plus some omitted variables bias. And so what we want to do is we want to think about when we construct g tilde this way, what bias what's this correlation, right? So this correlation between g and g tilde we saw before is the relevant thing for thinking about how much heritability is underestimated. So then we can just sort of think about correlation between g and the sum over j of these things, right? So these coefficients times the x's, right? Okay, so that's I don't have any sort of general results about that, but the way I was thinking about this is let's think about some extreme cases, and this is I hope these cases aren't far enough away from reality that they're useless, but I want to think about two sort of extreme cases, right? So one is, what if all the psi's are equal, right? All of these SNPs have the same effect, the same causal effect. They're all causal, and they have the same causal effect. So I know let's if that's the case, and all the correlations are the same. So we're not assuming no linkage disequilibrium, but just that they're all the same. Right? If that's the case, then we don't get any measurement error in the heritability. Right? The correlation between g and g tilde is 1. So that's interesting because we get bias in the GWAS, right? The estimates are biased, but they're biased, but that bias doesn't affect the relative size of the betas, and so we don't get bias in our estimate of heritability in the secondary sample. So another maybe more interesting case is, what if only one of the sizes is equal to 1? So we just have one causal variance, and the rest are all 0, but we have a bunch of correlation across the x's. Right? Then what we see is that this correlation, squared correlation, looks like this. Which is a little hard to sort of think about how big that is. But one case where we can analyze that is if the rows are all the same, and we think about the limit as j goes to infinity. So j is the number of SNPs, right? Then this is going to converge to rho, that correlation. So if there's one causal variance, but all of the SNPs are very sort of have a very small correlation with each other, this is suggesting that we're going to get a very big bias in the estimate of the heritability. So, on the other hand, if we actually other thing we can see from this formula, well, not quite from that formula, I guess from this one here, and it's more general than this case as well, but we get two special cases also where there's no bias. If all the rows are 0, then we don't have a problem. Also, if all the rows are 1, or in the limit where they're all 1, they're all perfectly correlated, then we don't get a problem either, right? So we don't get a bias either because it doesn't matter which ones are causal; they're perfectly correlated, and so we pick up the right signal. So generally, we're somewhere in between, and this is just giving one case of thinking about like what the bias might be in estimates of heritability. Okay, so I think this is the case that's a little more interesting to some of the economists. We want to include the PGI as a control in a regression. Maybe what we really care about is alpha, right? What's the effect of some particular environmental factor controlling for possible correlation with genetic components? So something like parental investment or something might be correlated with genetic components, and so we want to control for that. So just a little bit of notation. We can write these G and G tilde as this is just condensing the notation. So psi is this vector of all the psi j's, and beta is the vector of all the beta j's. Then we can sort of have a few results about sort of what bias we get in estimating alpha 1 here. So this is making no additional assumptions. This is just the using the FWL theorem, or what some people call regression anatomy, or whatever. So this is just a straight omitted variables bias formula. So we can think about some special cases again. So so this is, I don't know if this is standard notation. I think this is just notation I made up. If we do a linear projection of E on G and G tilde, and the coefficient on G is equal to 0, right? In other words, if controlling for G tilde is sufficient in a linear regression to explain the very relationship between G and E, right? So that's a pretty strong assumption that G tilde is a sufficient measurement of G in that specific sense. But if that's the case, then we get 0 bias in our estimate of alpha 1, right? So one case where that happens, the most typically basic one case where that happens is if beta is proportional to psi. So beta is the estimates we get from the GWAS, and psi is the actual causal effects of each gene. So I think this is helpful to understand, helpful point to understand, because they don't have to be equal, right? Maybe that's obvious, but if in the GWAS there's some other effect that we're not controlling for, so we don't include family design in the GWAS, and the bias we get in the estimates because of that for each beta j are proportional or roughly proportional, then this is suggesting that we don't get much bias in the secondary study, right? In our estimates of alpha 1, we would get big bias in the heritability estimates, right? Because we're attributing some of the variation due to correlation with parents' genetics, for instance, and the effect that has on phenotype to the individual's genes, right? So we would get biased estimates of heritability, but not biased estimates of this coefficient on the environmental factor in the regression. So the other important point is that this is not always going to be attenuation bias in this regression. So this factor in parentheses under certain conditions is going to be between 0 and 1. So we get attenuation bias, sort of natural sort of bias in classical measurement error, which is that factor outside of the parentheses is an omitted variables bias if we just don't include G at all. And so we get some fraction of that omitted variables bias due to not including G, not controlling for G. Some fraction of that is still there if we don't fully control for G. But that is not always going to be between 0 and 1. So it's not always attenuation bias. And then some important cases, we can go through some examples to think about when it is attenuation bias and when it's not. So one case where it is attenuation bias is if E depends on the SNPs in the same way that that Y does, right? So if one of those SNPs affects is correlated with the environmental factor in a way that's very different from how it's correlated with the phenotype that we're looking at, the Y we're looking at, relative to the other SNPs, then this is not going to hold. And generally, we're not going to get just a standard attenuation bias, and we would have to think more carefully about the nature of the bias. But this linear projection of the environmental factor on the SNPs is proportional to psi, then we get this standard kind of attenuation bias that depends on how highly correlated G and G tilde are. Another interesting case where we do get attenuation bias is if we're missing some of the SNPs, right? So if beta J, in other words, if beta J is 0, so we're not weighting it at all in our PGI, but it does have a causal effect. So it is a causal variance. And that's the only difference, right? And that's the only difference between G and G tilde, right? It's just because we left out the SNP or several of the SNPs. But the betas on the rest of the SNPs are the right value. Then we get attenuation bias. So a case where we so kind of counterintuitively, a case where we don't get bias is if our PGI includes too much stuff. So you might think that's sort of the standard measurement error story, but we have this extra measurement error in our PGI. We have some SNPs that don't have a causal effects that we're including, but G doesn't include. Like G has 0 coefficients on those SNPs. Because of linkage disequilibrium, because they're correlated, the SNPs are correlated with each other, and because these extra SNPs that we're including might be correlated. They might not be causal in the for our phenotype, but they might be correlated with the environmental factor. Then that causes bias, and that bias is not generally going to be attenuation bias. It could be, it could be a positive or negative bias. How am I doing on time?
You have more than 10 minutes.
Oh, okay. Okay, so then, so the next thing is to think about this model with an interaction effect. So there's a very easy case and a case that was that I didn't sort of drive any general results for. So the easy cases of G and E are uncorrelated. And actually, I thought that was not an interesting case, but after yesterday, it sounds like that's sometimes what we want is for G and E to be uncorrelated. If that's the case, then the bias that we get is basically just this attenuation bias due to correlation between G and G tilde not being 1, right? Which is not to say that that's not important, right? That's that correlation could be fairly small, and so that would bias the estimates of G by E effects towards 0. But if G and E are correlated, then again, it's hard to characterize in general what kind of bias we get on that beta 3 term. It's not just necessarily bias towards 0. Okay, so what about some possible solutions? So one thing that I've seen there's a talk yesterday talking about this, and before I think it was in Kevin and Pietro and co-author's paper, suggesting that the solution to this measurement error problem is to use instrumental variables. Use 2 different independent samples to construct 2 separate PGIs and use one as an instrument for the other, and then reverse that and do the opposite. Use one as the variable and the other as the instrument. Which is my understanding of what this OR IV is. It's basically doing IV twice both ways. So that is going to help if the measurement error in those 2 PGIs is independent. But if the measurement error that we're worried about is this measurement error due to linkage disequilibrium and the biased estimates we get of the betas of this size, the coefficients, then the bias we get in the first sample is going to be similar to the bias we get in the second sample, right? If they're and so they're not going to be uncorrelated, right? So they're both overweighting one particular variant, for instance, not because of sampling error, but because of this omitted variables bias. So the instrumental variables doesn't solve that problem. Maybe that's obvious. I just want maybe that, but was one thought I had. So what are some other solutions? So as I was putting these slides together and doing more reading over the past week, started to see that, obviously these are not issues that no one else has thought about before, and there's a lot of work in statistical genetics by a lot of people in this room that possibly address some of these issues. And so it's unclear to me, how much how much we can use things like LD score regression and genetic correlations to solve this problem if what we're interested in is estimating these kinds of models that we saw the presenters yesterday estimating. There's this method, I think James or Q, one of you two, talked about LD-PRED a little bit earlier today, that's supposed to sort of address this problem by modeling the linkage disequilibrium. I think that's that's something that's I would like to understand more about, because I think that's possibly a reason why everything else I already said is maybe close to a solved problem. But I think my understanding is that those methods rely on some underlying assumptions about the nature of the linkage disequilibrium that may or may not be true. So when I Googled LD-PRED, the first thing that came up on the Google search was some abstracts or some paragraph from a paper that said, LD-PRED does this, but there are a lot of limitations of LD-PRED. So my takeaway from that was that maybe this hasn't solved the problem completely. And then another solution is, why don't we just estimate the model that we want in the original sample? And I think there are reasons why not to do that, because we don't have, like Q was saying, we don't have one big sample. We actually have all these different cohorts that they're combining with meta-analysis. But if we can figure out a way to do that, and actually, I think there is some recent research sort of thinking about how to do that, then that would bypass these issues. What else? Okay, so the last thing I want to talk about then is this possible sort of relationship with these ideas from the literature on test score, some of my work. So when we think about a test, right, we give a bunch of questions to somebody. It could be like an IQ test or an AFQT test or a Likert scale, where we give people a bunch of statements and they agree or disagree. Anything like that. And each response is MIJ, so individual I responds to each of these J items. And often what we'll do is we'll construct an average response, so the sum of the responses across those different items. And sometimes we might do that with some weights, but often we don't include weights. It's just an unweighted simple average. So some of the work that I've done is sort of provided conditions under which this is justified if what we're trying to do is control for latenability. And the main assumption that's required is that these items are independent or weakly independent, conditional on some latent variable theta, right? So some latent factor, latent ability, and potentially that could be more than a scalar, but there's some lower dimensional skill or ability that we're measuring with these items. And sometimes we'll choose weights to mitigate the bias, but the bias goes away as J increases. Right? So this is actually very different from the PGI scores, right? So what I think is the easiest way to see that it's different is there's a link between why MIJ affects some outcome and why MIJ is correlated with other items, right? Why a particular item is correlated with other items. And it's that link that makes this, that justifies this, right? So we don't think that there's some latent. I don't think that there's a biological basis for thinking that there's some latent, lower dimensional construct that actually sort of causally affects the genetic data, right? And that's why the genetic data is both correlated with itself and has impacts on phenotypes. So I don't think there's a reason to think about the PGIs in that way. But then the other thing I was thinking about as I was working on this was maybe this suggests that we shouldn't be doing this with test scores either. Maybe each item on the test is actually measuring a different, slightly different skill or ability. and it's that combination of those skills that matters, not some latent thing that's. And so, I think a lot of times we, or maybe I, have taken this kind of model for test scores kind of literally, and sort of, this has caused me a little bit to reassess that. That's so. So one thing since I have time, I have to. A little time. Okay,
5 minutes.
one thing I wanted to go back to actually was this. I don't know if I have a slide on it, but so there's this issue of using like regularization methods to get better estimates of the coefficients here, right? The beta hats, right? To not just use linear regression, but to use lasso or other regularization methods. So I think one thought that I had on this, and I think actually John alluded to this as well, is that there's work in econometrics by Chernozukov and others, thinking about this setting where we're not trying to predict something, we're trying to control for something, and how we do lasso is different in that setting than if we're trying to get the best prediction, and that's generally going to be true of a lot of these like regularization methods. So I think one thing that would be interesting is some of the stuff that Q talked about, right? The goal there is to get the best possible prediction, and like my very basic understanding of a lot of these regularization methods is we're allowing some bias to reduce the variance, but it's not clear that that's the trade-off we want to make when we're trying to control for that in a regression. And actually, it's some work by others has shown that the way we want to do that is different if our goal is to get the best possible, right, to minimize measurement error when using this as a control variable. One other thing that and so then there was also this question about just looking at my notes here. No, I think that's it. That's that. I think I already talked about that. Okay. So that's all I have. Thanks.
Then you use the mics because Patrick Turley is on Zoom and he's probably the leading expert on these measurement error issues and PGIs. So Patrick is actually the lead author on paper that deals carefully with the issue of measurement error in the betas. I think that what you said, that basically LD-PRED essentially solves the problem, is essentially right, with one subtlety, which is that, as you also said, it's if you could use multivariate regression, and control for all the SNPs, then you would completely solve the problem. Talking about the problem of the bias correlation between the genotypes and the error. So multivariate regression would solve the problem. LD-PRED doesn't, still allows a bit of bias, but what Patrick showed is that the bias is very small when you actually run the regression using the PGI from LD-PRED. And I guess I'll also mention that paper develops an alternative method for correcting for the measurement error part. That's different from ID. It's essentially just a disattenuation approach. We have estimates of the SNP heritability, so we can directly apply analytic formulas to disattenuate coefficients from the regression. And I think that's generally going to be better than the ID approach because it uses more information or data.
Should I stay up here?
Wherever.
Good. You have a comment? Right. Oh, I have a comment, but not related to this.
Oh, okay, yeah, David, you're.
Oh, no, I just wanted to clarify one thing. I liked the talk. I was very impressed by how you just managed to figure out a lot of the key issues that people wrestle with in practice within these applications. So the issue isn't that that no geneticist has ever heard of the Fish-Lau theorem. The issue is that you need this expand X inverse matrix to apply it, right? And so in practice, what people do is they use an approximation, and there are two sources of approximation error. The first is that you have to use some block diagonal approximation just because of you don't have the data. And the second is that you have to estimate it in a different sample where the variance covariance patterns might not be identical to the one in the sample that you're interested in. You just don't have that information. And to see how things can go wrong, imagine the two-snip case. Then the formula that takes you from the univariate to the multivariate coefficient has a term that's 1/1 minus the R square of the regression of one snip on the other one. So imagine that in your sample that you're using to estimate this pattern, maybe that's R square for some snips is 0.99, but the true R square in your estimation sample is 0.9, then you're going to be off by roughly a factor of 10. And that can be a huge source of problem, practical problem.
There you are. I really enjoyed the for a few reasons. So I'll just say one reason and then I'll mention or highlight a sentence. So the idea of like using latent factor models or some kind of something that will reduce the dimensionality or to get at a component and then shovel the rest to a measurement error versus PGS. Yeah, it's kind of like almost opposites in a way. Like we're trying to get as much variation in all the different sources and not the common component, but like all the things that might contribute to education and all the different pathways. Whereas, so I don't know, that was clarifying for me. And of course, those raise the question of like maybe we should treat each test scores different. But I don't know. In some cases, but the thing that you also said, I think somewhere was, I don't know, particularly words into your mouth, but a disconnect for me sometimes, and I think you articulated it, is that sometimes the people who have the most expertise or the behavioral geneticists are solving, working on issues that aren't necessarily the issues that we need to work on, right? And so, I think that's maybe where there's a little bit of room for some kind of collaboration, and that, we're terribly interested in things like, gene by environment interactions when the polygenic score was measured in some kind of a specific environment. For example, if that's true, how is that going to lead to a bias in the estimate, right? Among a lot of things. And I just think that perhaps we should start writing down that model, and then run it by the school of SSC for doing it right. But yeah, I think that it would be useful for us to actually because I find myself kind of relying on results from people who are more deeply involved in statistical genetics and trying to sort of fit it to something that I'm worried about, and it's not always the case that I can. So.
Actually, I want to amplify something, just riff on something about this related too. Is that I feel like in an actual applied settings, we need a lot more like second best practices, because like if we want to do G by E or any type of analysis, right? It's like usually we need to go to specific data sets that have really rich data on mechanisms or environmental contexts or beliefs or whatever it is that your specific question is. And so the answer, it's unsatisfying to be like, well, the answer is you just get a better data set, right? And I think that happens sometimes with like a lot of the work that is very heavily focused on the UK Biobank. It's like, well, there's great things about the UK Biobank and having trios and having large sample sizes and doing it GWAS in there, right? But there's also very limited, data on the kinds of economic outcomes and behaviors, et cetera, that you really like. And so I think it would be really great to be like, okay, I'm stuck with, I'm stuck with the HRS, right? And how can I kind of leverage insights from different, GWASs that have corrected for some of these problems to at least characterize the bias or the problems that I'm going to run into with my second best world. I mean, otherwise, we can't ask those questions. Right? It's just riffing on it. Sorry. Yeah, Jim.
Let me just ask Ben. Follows up. I like the discussion a lot. But he suggested something I've been thinking about a lot in this context through this conference. So the factor model was immediately dismissed as kind of a way to sort of unify these different XIJs for me. But why is that true? I mean, there seems to be this pattern, right? A very high level of correlation among these X's. So wouldn't some kind of version of a factor model, a discrete factor model, some kind of classification model work? I just wonder if it's been tried to have a latent variant. It's almost going one step deeper. where you're saying, I have these factors that are generating these X's, but they're low dimensional. That was kind of dismissed out of hand. I don't know if that's true. I'm just wondering if it's been tried, and yeah, please.
Sorry to promote myself, but, so yesterday morning, or maybe the evening before, I posted a preprint. So you raised the question of, do these, I'm also actually very interested in response theory and measure it. Raised the question, do these thetas really exist? So this preprint was about neuroticism, a 12-item neuroticism questionnaire administered in the UK Biobank. We basically asked the question, with genomic SEM, if we do a factor model where we say these 12 items are indicators of a theta neuroticism, and we say that the SNPs associations with the 12 items can be explained completely by them acting on this hypothetical theta. How does that fit versus a model where we freely constrain, unconstrained, the SNPs associations with the 12 items to be whatever, opposite sign? Basically what we found is that, well, not with one theta, we have to have two additional factors to satisfy completely the principle of events, but maybe surprisingly, we do find SNPs that look like that, a lot of them, that look like their associations with the 12 items are explained by a neuroticism factor or theta actually existing, and they're acted on by the SNP. So
You sent a paper around.
I think one thing that one of my thoughts on these this is that in the IRT model, like the test score, like I said, the reason that there's correlation among the items is the same reason that those items affect the outcome. And my understanding is that a lot of the genetics literature is assuming exactly the opposite, that the betas are actually uncorrelated with the LD score. And suggesting that, like, you know, if we just look at right, and this is why we get biased because of LD, because they're not perfectly correlated there. I don't know. I think didn't mean to dismiss it out of hand, the idea, but just seems like a very different reason. If you would use these methods, there have to be a very different justification, because
It's just the data reduction scheme.
yeah, exactly.
So you're basically just taking the X. I'm not sure you're making a strong position in terms of betas and X's and that side. You're gonna try to find a new set of betas, factor one, let's call them, associated with the extracted factors.
No, what I'm saying is maybe these
Possible interpretations.
SNPs are all highly correlated, but only one of them is positive. Right? That's not the way we think of test scores. Only one of these items actually matters for wages. The rest of the questions don't have anything.
Well, I think we use just these multiple questions to try to get a reading of this latent score, call it IQ or some measure of ability. So it's a latent factor that's underlying.
Yes.
It's generating that correlation. That's the whole factor.
Can you understand what you're thinking about the factors and variation in the SNPs?
Yeah.
Well, so there's a few comments. First, we do standard genetic analysis. We take the first principal component of the whole SNP data, but then we use these as controls for population stratification. But the reason why we don't use these factors and just regress, DAs on all the pieces basically, is because they don't really capture like common variation in the genetic factors. Think about it this way. So first correlation across chromosomes is 0 for the sort of related, like first order of destination. So and also these big factors, all the 1,000 first factors will just capture big axes of ancestry dominated by, genetic drift and other factors rather than the two factors that vary in terms of their causal effect on indication.
You get both sets of factors work. Those are mutually exclusive.
But how would you assign these factors for the SNPs that matter for indication of pain?
Well, again, we asked the question of which ones are predicted. You have this data of education, a bunch of different measures of education. And we ask this, which of these factors is most predictive? Well, in fact, for a lot of these are most extractive factors. That may not be these biggest factors.
The factors, though, but the thing is, because any factors
That's
that
kinda like what you-
across the genome, they're local to the genome in the sense they don't correlate across chromosome or.
It's, I'm just looking for how many components there are, like,
Well, so there's LDB1 is one thing that.
that's
like-
less constructing factor.
Yeah, well, maybe we're talking about two different things, but
No, but
I mean, some geneticists have created data sets of LB blocks, which are chunks of the genome that are typically put together, but it's just a fact of like having strong correlations with.
right, that's what, that's kind of the metric I'm starting to do. You gotta have a local version of that, wasn't pieces of genome.
Yeah, so I guess we could correlate EA on each of these LB blocks, but I think a lot of the household SNPs are, those tiny. I don't think it might be better. Anyone who starts something.
It's just a question, I wasn't telling to do it.
No, it's interesting, and then for me to think.
No, I just think it seems like a natural. Then just dismissed it out of hand. I thought, well,
No, I didn't mean to.
I just wanted, why we would dismiss it, that's all.
You can imagine a model where some SNPs impact education through, say, IQ, and some SNPs impact it through other channels.
Correct.
And people have done that, and this paper in Nature Genetics that's, uses this essentially GWAS results in two separate analyses and relies on one of these psychometric
And then you have
models.
vector problems, like you just.
Oh, sorry, but
You could have like vectors, or.
You could have something more.
You have a vector effect, there's not a scale effect.
Oh, sorry, didn't know.
Sorry, James.
Yeah.
I wanted to understand, so what you're saying is not that we should try to extract a single factor and all the G's are some measurement of it. What you're saying is that, and if you do kind of like locally, you're saying, okay, it's IQ and also like having dimples are the two things that make you do well in school. And there's a bunch of SNPs that do the first one and then a bunch of SNPs do the second one, and those are the two factors that you try. Is that the idea? That you're kind of thinking about?
Were you talking to me?
Yeah,
Start.
I mean, I'm
Yeah,
looking
no.
in your direction, so I mean, anyone.
Get some data reduction.
I see.
So the fact that different SNPs are, see, I don't know quite what adjacency means biologically. I mean, you're assigning a lot of weight to them, I'm not exactly sure what it means. And is it that they have the same neural tissue, they sort of overlap in some fundamental biological sense? Because right now it sounds like just a statistical correlation, so we're approaching it that way, not.
Pietro's got it.
I mean, I think that one of the points that people have been discussing about it, that I think that the correlation between SNPs is not informative on the effect that SNPs have on outcomes. And so a latent model that assumes that there's a latent genotype that is explained by the correlation among SNPs, and this latent genotype is the one that influences the outcomes that we care about. It doesn't seem to be a good fit of the data, rather than a model where we say each SNP has its own effect on a latent phenotype, latent outcome, that then has an influence on the measurements that you have.
So they just maybe through local, right? So
But exactly, so if you look locally, so when we say that the SNPs are close to each other, it means physically close on the chromosomes, and one next to each other.
Yeah.
And that, the fact that they're physically close to each other doesn't necessarily mean much, if not a heritage of our ancestry.
No, I just want to use this as a tool to extract whether it means much or not.
Yeah, so
I'm
it's
not saying it does, I'm saying
Yeah.
it'd be kind of a way to kind of initiate a search.
So I think that people have tried to use that correlation, and the only thing that came out of that correlation is your ancestry. Nothing about the way that genes influence the outcomes that we care about.
So within the UND,
Empirical.
blocks or factors.
John, it's,
Numbers have decreased it because of the effect of blocks. I mean, causing good washout. The causal effects of the care.
I'd just like to raise a question that, what I see a lot, is just constructing an index, and if you don't get the SNPs quite right, as long as they're nearby, that's good enough. But it would increase my confidence a lot if you could say, let's take cases where we know genes do something like cause your hair color. It's been independently identified. Can you do a GWAS and not only just get the nearby, but actually get close or nail the SNPs that cause hair color? And if I saw that there and say, wow, I'm believing this, right? But if they're not working very well, then I'm wondering, you've got a lot of measurement error there and they don't really do a good job of identifying the genes.
But this empirical Bayes method that you heard earlier, we try to isolate that. Giving a lot of point tests on that gene, right?
The point of that is to still improve predictive, not to somehow improve the quality of hypothesis testing.
But you could use it.
You could, but I will actually have a separate comment, but I wonder maybe it will be highly correlated with what Dan is going to say. But people have tried exact that idea actually on phenotypes that we think we understand a little bit better. Things like urine, you understand a lot of biology about why urine levels differ between individuals. But then when you have a huge sample size, you still end up having this omnigenic, polygenic genetic architecture. You have signals everywhere. It's a $1 million question in human genetics why you see that. But the good news is the top hits do tend to cluster around the expected genes you want to see for this phenotype. But still, if you aggregate those, what we call core genes, in terms of the amount of variance these genes explain in the population, it's actually on the lower. It doesn't fully explain the total genetic contribution to urine. You get a lot of baggages coming from elsewhere. Those are the stuff that you previously wouldn't expect before running the GWAS. But then people have come up with theories to explain why you actually see that. So one of the popular models out there is called omnigenic model, which is saying that, okay, you have pervasive, regulatory effect across genes that are expressed in the biologically relevant tissue. For the phenotype you want to study, for EA that will be brain, for something else it will be something else. But then
Not for hair color.
for
Hair color is just a few genes.
no, I don't know about hair color, but for hair color, there is a Science Advances paper that gave you, I don't know, 50 different gene-wise hits. So I wouldn't call that a monogenic trait. But if you look at the textbook for eye color, you have those 1-2 genes that are presumed to be the mutation that originated at some point, but eventually became fixed in the population. But if you look at GWAS signal, that's simply not true. You still have a lot of variability. Explained by genome-wide SNPs. What I was trying to get at is the short version is people think some dense regulatory machinery behind how gene expression works, how they interact with each other, how certain genes influence other genes expression is the reason why you get signals everywhere. So you will have some master regulators in the gene expression network. The strong EQTL, strong SNPs that influence that gene's expression will affect gene expression of a lot of downstream stuff, and then each of them will have direct, indirect effect eventually on the phenotype. If everything is active in that relevant tissue or cell type, eventually, when your sample size is big enough, they show up as some kind of weak signal. But because this type of weak effect is everywhere, altogether they actually explain a bigger chunk of R-square, bigger chunk of heritability in your data. The core genes in terms of R-square is actually on the lower.
I'm gonna just intervene here that we need to get to lunch. But Dan, I know he had his hand up. Do we have
I think it's getting.
is it covered? Okay, then good discussion. Let's get to lunch.